Thank you very much, everybody, for joining for this workshop. My name's Graham Murray. The program has promoted me. I'm Dr. Graham Murray, even though the program says I'm Professor Graham Murray. So I'm Associate Professor in Cambridge. And I'm going to talk about causality in psychiatric research. And I'm going to talk about brain imaging, because I know about brain imaging, but actually a lot of the points I want to make would be equally applicable to any other biomarker. But because I know about brain imaging, that's the example that I'll give. But before we get into the brain imaging, let's talk about another example of causality. And this is if a patient goes to the doctor and says, well, you know, on Monday, I had gin and tonic. Oh, sorry, the graph hasn't come out. But on Monday I had gin and tonic and then I felt sick, okay? And then the next day I had vodka and tonic and then I felt sick. And then on the following day I had whiskey and tonic and I felt sick. So we can draw the conclusion that tonic is very bad for you, yeah? So we can do that, can't we? Because we've done an observational study and we're drawing a conclusion. Of course, we can't do that, right? We know we can't do that in this example. But we forget that when we spend millions of pounds doing research sometimes to collect a biomarker. Okay, but that's not to say that all the observational research isn't important, but it is important. This has been a very important and influential paper. So this was the first sort of modern brain imaging study. Eve Johnston, Tim Crow, Chris Frith, and colleagues. And this is a photocopy of this Lancet article, actually from the King's College Institute of Psychiatry library. And this was in the days when it was a real journal, and this just shows you how much people read that article. I'd be amazed if anyone ever read any of my articles as much as clearly that has been read, and so much so the page has been ripped out. That's this slide actually Ed Bullmore made taking this photograph. It's a very influential study. 17 patients gets you into the Lancet in those days. Not anymore. This is a brain. This is my brain. So the article's about the ventricles. If you're not an imager, these are the ventricles. This is a so-called T2 weighted scan, so water comes up white. So these watery areas in the middle here are the gaps in the brain, the ventricles. And this paper's about ventricular size. And you can see there's a difference in ventricular size in the patients and the controls. The patients in open circles have had surgery to their brain, so that's a possible confound, but nevertheless, you can see there's a group difference, although some overlap. Oh, and this paper, it tries to look, not only do you look at case control differences, but tries to look at relationships with some clinical features, which of course is difficult in a small sample with variation. But nevertheless, I think it's very difficult to really conclude whether, do these differences in the size of the ventricles, are they what is causing the illness? Or maybe is having the illness leading to certain behaviors that then cause a brain change? Or maybe there's a third thing that causes one to have a particular brain feature, particular ventricular size and a particular illness, schizophrenia, but they're not really correlated at all. They're just sort of existing side by side. And an observational cross-sectional study like this doesn't get us to causality. It's interesting, but it doesn't get us to causality. well, maybe, so that's my first conclusion, is cross-sectional imaging studies don't tell you much about causality, but, it'd be the same for a blood test study or a cognitive study or any other study you wish to do. But maybe we can get to longitudinal studies. Maybe longitudinal studies will give us the answers that we need. And this is a longitudinal study that I was involved with, which is a collaboration between my department in Cambridge and then my former department in Finland, in Oulu, where I used to work, did my PhD in northern Finland. And as you know, very good health records in Finland. So we're able to identify all the people who've been diagnosed with schizophrenia and access their health records and scan a subsample of those. So we identified all members. There was a birth cohort of all the people who were born in the northern part of Finland in 1966, which was about 10,000 people, and of those, over 100 developed schizophrenia by sort of early adulthood, and of those we managed to get baseline scans on something like about 60 or 70 and then repeat scans on about 33. So, it's not all the patients, but we know what proportion of the patients it is. So there's some attrition over time, of course, in not being able to follow some people up. So we've got about 30 patients, just over 30 patients, and about 70 controls. The good thing about this study, the controls are randomly selected from the population, not just inviting all your friends and your PhD student to come and be in the study. So that's good. And so this is over the 30s, being scanned at baseline early 30s and follow-up early 40s, about 9 years interval. I was about the same age as this study. I was about the same age as the participants when we were analysing it. So I was interested in the results. So let's look at the controls are on the right. So this is now a T1 scan where the dark on the picture is the water. So here are the ventricles in the center. And you can see this is baseline and this is follow-up. We wanted to save time in the follow-up, so we took quicker pictures, and that's why it's more grainy. We had less scanner time. So you can see that controls, as you go through your 30s, here's one healthy person who has a more watery brain with bigger ventricles when they're in their early 40s compared to in their early 30s. So this is mild atrophy, more prominent and then more substantial. Atrophy, it's health, this is healthy atrophy. It's happening to us all. We can't do anything we can't do much about it. Maybe you could do something about it, but it's just, as we age, our brain is atrophying. Or we're getting less of it anyway, and more water, less brain. But somehow we managed to muddle through. So these are controls showing differences. You do not need to be a neuroradiologist, do you, to see this? I was quite shocked, actually. I had not really, I thought, maybe when you get to your 50s, as I am now, but in your 30s. Okay, so this is the controls. And with the patients, it's just the same pattern. It's a little bit more prominent. This is the percent brain volume change. So 6% loss in this person, about 3% loss over 9 years in this person. So just some examples of patients who, same aspect as the controls, but a little bit more prominent. Here's one patient who quite more larger ventricles. You can see the gaps in between the brain folds is a bit more prominent at follow-up compared to at baseline. And so the yellow is just pointing to areas where you see there's been some change. And here's a graph showing the individuals and just showing there's a fair bit of variation in the controls. on the right, and there's a fair bit of variation in the patients, and there's an overlap between the patients and the controls, but on the whole, the patients are on average, the brain is shrinking by about 0.7% per year, whereas in the controls, or 0.6% in the controls, it's about 0.5%. So, what do we make of this? Is this shocking? Is this trivial? Is this important? One way of looking at it, so at the time, people were very interested in whether medicine could be doing this. Maybe this is something intrinsic to the illness process. Maybe this is something doctors are causing by prescribing harmful medicines. And so a PhD student read through all the notes of all these 33 patients and very carefully documented, there's 10 years of records every time a medicine was prescribed. And we knew it was being prescribed and issued. We had the pharmacy, we knew from the pharmacy it was being issued. We don't know if it's been taken, but we know if it's been issued. And plotted on the x-axis that over here is taking more antipsychotics over time. And then this is the percentage brain shrinkage. So the patients who are taking more antipsychotics over time are having more shrinkage of the brain, which might make you conclude that, the medicine's causing it. But the other thing I can tell you is that the most poorly patients, what does a doctor do if a patient is not very well? Most doctors just put the dose up. It's not very sophisticated medical practice, but there's a natural tendency to do it. So it's very difficult to disentangle. Maybe just the sickest patients cause the patient's not doing very well. I'll put the dose up, and so maybe this is not causal. Maybe it's just this is sort of more severely unwell patients tend to have more brain loss over time, and that happens to coincide with patients that the doctor in desperation keeps putting the dose up even though it doesn't do much good, probably just causes more side effects sometimes. But we want to try and be helpful. And here's just another way of looking at that, sort of plotting at patients who were taking more medication, controls, very little medication, and in the middle, and this sort of seeming sort of response with the group that's prescribed the most medication losing more brain volume. But initially I thought this was a great cause for concern, but then I realized once you put into perspective that it's really that all of us are losing the brain over time and somehow we manage. I think it's just a very crude measure, how much brain you're losing, and in itself it's just a lab test, not necessarily important. What's important is the implications for how one lives, one's function, and so forth. But we still haven't got to causality. So we've done a longitudinal study now, took years to do, a lot of time and effort and so forth, and we can make some associative statements, but it's still very hard to get to causality. And again, I don't think, it would be the same with any other biomarker. So how are we ever going to get to causality? Just to say, it's not to say it's not important. And I mean, I've done a lot of these observational studies in brain imaging. This is one of mine, brain areas associated with being a people person, correlative study, that's one of mine. Here's another one of somebody, this isn't one of mine, but this is looking at relationship between air pollution and brain changes. And you see this sort of thing. Anton earlier, Professor Gorek showed one about TV time and brain structure again, So there's all these associations, but where's the causality? And here's a book about this. It's a good book if you want to read a popular science sort of account of whether it really matters, whether you're losing brain. Should we be concerned, essentially? The strapline on the back of this book says, your brain is shrinking, does it matter? And so if you want to read a popular science account of this, I highly recommend this book. It's actually just been reissued with a slightly different title, Rethinking the Brain. and a picture of my brain's in it, so I've got an interest in it. Okay, so how are we going to get to causality? Well, we get to causality through trials, don't we? That's how we get to causality, through RCTs. And we know that because we randomly allocate the treatment, then we randomly allocate people to a treatment group or a control group, and then we follow up. And then we know that any difference here must be caused by this. Because everything else, if you've got a big enough study, all the other things that you can't measure, all the unmeasured confounding factors that might predispose somebody to behave in a particular way and might influence an observational studies, basically all the stuff, like we know that the tonic was not what is making the patient sick, but that's because we already understand the effect of alcohol and tonic on the body. But when you're trying to work out causality, you don't know what is a confounder and what is causal. And randomization just takes care of all of that unmeasured confounding. But we can't always do an RCT. So what can we do? We can use genetic information in a technique called Mendelian randomization that some of you may already know about. So if you already know about it, you can just switch off for a minute. If you don't know about it already, then I'll have a crack at trying to explain it. So just think of it as a natural equivalent of an RCT, a sort of a natural experiment. In the population, genetic variants are randomly distributed across, we all know we have these genetic variants, and they're randomly distributed across the population. And just as in an RCT, we have a control and a treatment, in geneticists always call the sort of the average, the normal one, or the standard one, the wild type. And then you have a variant to compare against that. So that's exactly the analogy of a control and an intervention in an RCT. And then if you look at the outcomes, then we know, and we look at them in relation to these genetic variants, then we can draw a causal inference, just as we can draw a causal inference in a randomized controlled trial. And I'll give you an example. But so when can we apply this model? That's really the thing we want to know is when can we use this technique? Okay, so we want to use the technique, just to recap, because in observational studies, we're going to have some unmeasured confounding. There's sometimes some measurement error. We can have, it's difficult to know the direction of causality, so you can have reverse causation. But Mendelian randomization, I think somebody earlier said they were an economist. I don't know if they're still here. But in economics, people use instrumental variable analysis, which is a general technique in statistics, and this is an example of instrumental variable analysis where the instruments are genetic variants. So we're using genetic variants as proxies for environmental exposures, and this is robust to many of the limitations of observational studies. So just as in a randomized controlled trial, randomized assignment of treatment evaluates the effect of the treatment on the outcome. In Mendelian randomization, the genetic variant is considered a naturally occurring form of randomization. So I'm going to give an example that I think is the best example for getting your head around it if you haven't come across it before. Do we have any so it's traditional at this point, isn't it, to ask a medical student to answer the question, but medical students usually know this because they have it in their exams. So I don't know if we have any medical students. No medical students, okay. So anyone want to have a crack? Anyone remember how alcohol is metabolized in the liver? What's alcohol metabolized to? Acetaldehyde, yeah. To acetaldehyde, and then on to acetic acid. Yeah, very good. And the key thing is acetaldehyde dehydrogenase here. And as you know, some people are rather intolerant of alcohol and feel quite sick, even in small amounts, and have a facial flush. And this varies by ethnicities. It's more common in some ethnicities, but the variation is there within all ethnicities, or within many ethnicities. So we know that there's this genetic variant that's actually unusual. So Isabel was talking about genetic variants, and usually there's a tiny effect. But this is a genetic variant that is common, that has a massive effect. And here's an example of how strong the effect is. So this is a study looking at genetic variation in acetaldehyde dehydrogenase. And let's just look at the men first. And the study is conducted in East Asia. And in the particular study sample, women didn't drink much alcohol at all. So it was mainly done in populations where just in those societies, women didn't drink much. So there's not much variation in how much women drink. But if we look at the men, these are people who metabolize the acetaldehyde quickly and don't get the buildup here with this homozygous representation of the genetic acetaldehyde dehydrogenase. And here are people who have 2 copies of the genetic variant that mean you get a buildup of the acetaldehyde. And these men hardly drink at all. And so here we can see that the genetic variant is a very good predictor of the behavior of alcohol consumption. And one might say, oh, well, hang on a minute, it's, maybe this genetic variant. I'll show the next slide first, actually, before I give that example. So, so we're seeing that in men, the genetic variant has an important effect on how much you drink. But societal and environmental effects are obviously very important as well, because it just was not customary for women to drink at the time. So there, the genetic variant is unable to take its effect. Oops. Okay, so now we're going to put alcohol to one side for a minute, and we're going to look at this genetic variant that we know is a good predictor of drinking in men, and we're going to see if it predicts blood pressure. And here's a meta-analysis of several studies looking at hundreds of people altogether in men, and showing that if you have the genetic variant that means that you have the buildup of acetaldehyde, then your blood pressure is lower. Okay, so having this genetic variant is associated with lower blood pressure than if you have the homozygous type that means that you can drink a lot. So we can draw a causal inference here about alcohol, even though in this slide we have not measured alcohol. So you don't have to know whether these people drink. We know that in men, at this time, these genetic variants are very good predictors of how much you drink. And then by looking to see whether the genetic variant is associated with blood pressure, we can draw an inference about alcohol intake. And one might say, well, hang on a minute, maybe this genetic variant is not acting through alcohol intake. Maybe it is directly affecting your arteries or something to affect your blood pressure. Well, in that case, it would also affect women, wouldn't it? So if the genetic variant was doing something to your arteries to affect your blood pressure, it didn't matter how much you drink. We'd see the same effect in men and women. But because in women, we know that the genetic variant does not influence alcohol. This is a nice control example here. So this genetic variant doesn't influence blood pressure in women because it's not acting through drinking, but in men it does. So by knowing the genetic association with the exposure, alcohol, we can then start to use the genetic variant to draw inferences about alcohol intake, even in studies where we don't measure it. So it does take your head a while to get around that. But if you just treat it as exactly analogous to an RCT, naturally occurring randomized controlled trial, then it can be very helpful in understanding how we can use it for causal inference. Okay, so can we use this framework to think about psychiatry? So give an example there about cardiology, but let's think about psychiatry. And I had a student a couple of years ago, Ida Sayed Salehi, who was studying public health. And at the time, there was a big campaign in the UK about can we stop dementia by maximizing brain health. And so we were interested in looking at some of the causality underneath this sort of idea. So we know observationally there are associations between brain size, brain imaging phenotypes, and dementia. We know that longitudinally, we've discussed before that controls the brain shrinks as one ages, but this is in Alzheimer's. At baseline, the brain has already shrank, and it continues to shrink as the patient ages. So observationally, there's an association, but what's the causality? Is the shrinking brain causing you to get Alzheimer's? If so, Anton's study about TV and the brain might be a bit of a concern. You could maybe think, well, if TV causes your brain to shrink by spending hours watching it, then, and maybe that puts me at risk of Alzheimer's, or if medicine really is causal, we don't know if it is causal, right? But if medicine or schizophrenia are causal for brain shrinking, and then is brain shrinking causal for dementia, so one can start to posit lots of causal chains, but can we test them? So Ida did this work that's actually just been published this month. So it's very topical, just in the current issue of the neurology journal Brain, if you want to read more about it. So we're going to look at the relationship between education and the brain and dementia. So education is a potentially modifiable risk factor for dementia. There's lots of observational studies that suggest that the longer you stay in education, the less your risk of dementia. But it's difficult to know, again, whether that's causal, because maybe the sort of people who are going to go on to get dementia are the sort of people who might leave school early. One could imagine lots of adversities in early life that might lead you to both of those outcomes, and actually it might not matter how long you stay in school. But nevertheless, it's an interesting and potentially modifiable risk factor. And one can postulate that this is presumably something to do, if it really is causal, it's presumably something to do with the brain, that education does something to your brain that protects you against dementia. So we wanted to test this model, whether education attainment has a causal effect on dementia, and assess whether this was mediated through brain health. And the way we could measure brain health was just through brain scans. I mean, there's other ways you could measure brain health, but this is a direct way. So if you like DAGs, epidemiological diagrams, directed acyclic graphs, then here's one. Educational attainment could lead to late onset to dementia. And could it be that this causality is mediated by what we've called imaging-derived phenotypes, but stuff you can measure on a brain scan? And here's some details of the study. We looked at quite a few different imaging phenotypes. So when you get a brain scan, you can analyze it in many different ways. You can look at volume, area, and so forth. So we looked at a whole load of brain phenotypes, but today I'm just going to mainly talk about volume and area. I'll talk a little bit about the hippocampus because we often think of that as very closely related to Alzheimer's. And when you do a study with Mendelian randomization, you don't need to go and recruit people anymore. You can do this on your computer because somebody else has already gone and done these big GWAS studies that Isabel was talking about this morning. We already know from studies in millions of people, actually, when it comes to education, the degree to which every genetic variant across the genome is associated with risk with years of education. So we already know that, and it's publicly available all across the genome, these measures of how much genetic variant A or B, and of course you can pay a company to measure your genetic variants. And we know how much all of them are associated with likelihood of having longer education. And then similarly, there have been some big studies of dementia, not as big as education, but with very large numbers, so we know the size, the effect size of a genetic variant, how much it's associated with dementia. And now we actually have big studies of brain imaging. So we looked at the Lancet 1976, 17 patients. Nowadays, we've got UK Biobank with 40,000, 50,000 people in a brain scanning study. So it's big enough to do a GWAS on and understand how genetic variants relate to brain structure. So those are our data. You just download your data from the internet. You've got your code, and you run the experiment to test your hypothesis. I mean, there's a bit of methods here that you don't want to look at every genetic variant. You really want to look at the ones that you're most confident about. So when calculating, when you want to look at the genetic variants that stand for your exposure, so here education, you want to use the ones that you're very confident really are associated with it. So we don't look at all the million genetic variants, we look at the 300 or 400 or so that we're very confident are associated with it. So that's the threshold here. And there's some other methodological aspects that I'm happy to discuss, but we'll press on. And you can plot this kind of a graph. So here in your study, what you do is you plot, so these dots are genetic variants that we're confident are associated with our exposure, which is educational attainment. And over towards the right are plotted genetic variants that tend to make you stay longer in education. And then here's the effect on the outside. The lower down they are, the more protective they tend to be over Alzheimer's. So the ones in this corner tend to make you stay longer at school and are quite protective against Alzheimer's. And on the whole, excuse me, there is an association. You can see it's not dramatic, but it is there. And the number of data points depends on the number of significant SNPs you have in your GWAS. So if you've got a small GWAS, you won't have a very big sample. And if you've got a big GWAS, then you can put lots of SNPs into your sample. So, just like when you're looking at an observational study, you should think, how big's the sample? When you're looking at a Mendelian randomization study, you think, how many SNPs were in the sample? And that could tell you about how confident you should be in the results. So we've got lots of SNPs that we know are associated. SNPs are genetic variants. So we can be quite confident. And one can do various kinds of, people often talk about, this is the standard analysis, inverse variance. What does that mean? It means that you are doing a regression and you weight the effect of each genetic variant by how precisely you can estimate its effect size. So that's the inverse variance, precision weighted. And you can do some sensitivity analyses as well. But that's the basic, you're just looking at the association between genetic risk of education and genetic risk of Alzheimer's. You see there's an association. What about the brain? Is the brain important? So here we have educational attainment as the exposure and brain structure as an outcome. And we can see that educational attainment, so this line is no effect. Here in surface area and volume, it seems we have a causal relationship, we have a genetic association which we can infer as a causal relationship between educational attainment and greater brain surface area, brain volume. No effect on cortical thickness and no effect on the hippocampus. Seems to be, there's a couple of folding effects, but we'll just focus on surface area and volume here. And so not that much effect on most of our imaging measures, but something there on volume and surface area. And this relationship might be reciprocal because you can look at the effect the other way around and you can see, does brain structure cause greater educational attainment? So we're just doing the reverse. Here, because the educational GWAS is big, we've got nearly 400 SNPs. Because the brain structural GWAS isn't so big, we've got much less. So, this is, maybe we can be less confident about the results. And the error bars are very big in some cases. But it seems that going to school, now having more surface area and brain volume leads to you staying to school longer. So it seems to be a mutually causal relationship that going to school does something to your brain surface area and volume, and that in turn maybe leads you to stay in school longer. So it could be this mutually causal relationship. There is another explanation, which I'll come to. But let's just look at the relationship to the brain to Alzheimer's disease. So here, there's really nothing going on. Look, everything is crossing the odds ratio of 1 and suggesting we can't see any relationship, any clear relationship between having any of our brain metrics as a causal factor or as an exposure and Alzheimer's as an outcome. If we look at it the other way around, what does dementia do to your brain? The main thing we find is that here we have hippocampal volume on the left and the right. Look, if you have dementia, your hippocampus shrinks. So we knew that already, right? We knew already if you have, but this is some evidence that really is causal. So evidence that Alzheimer's truly causally does shrink your hippocampus. Not rocket science, but that goes with what we know. But nothing to be concerned about here. There's no evidence that a shrinking hippocampus causes you to get dementia. So, if you read some study that something shrinks your hippocampus, you don't need to worry because, or at least if you're worried about dementia, it doesn't seem to be on the causal pathway. Another way of checking about causality is to look at the mediation. So what we can do is we can look at our effect of education on Alzheimer's and we can adjust for brain structure. So it's like adding that into the regression model and seeing if our effect size changes, and it doesn't change. So here's the effect size unadjusted, protective effect of education on dementia. If you add in all these different imaging variables, it makes absolutely, hardly any difference to the effect size. If brain structure was an important mediator, you'd expect that effect size to really change when you add it into the model, but nothing really happening there. So we're going to conclude that there's no evidence at all that brain structure, at least as measured here, so we had quite a few imaging phenotypes. Of course, there are many others one could look at, but as best as we can measure it here, no evidence that there's a causal risk between brain structure and dementia. No evidence for our mediation hypothesis. Some interesting stuff about education and the brain that wasn't really what the focus was, but we were, it's an interesting effect. Some caveats. The brain imaging measures, they're rather crude, aren't they? They're just pictures of how big the brain is or how much white matter there is or so forth. There's more to your brain than just those gross macroscopic measures, isn't there? So, maybe in the future people will look back and think these were very crude measures, even though they're state-of-the-art now. We didn't look at all the different tiny regions of the brain. We just looked at global measures. And the precision of these estimates will improve as the GWAS of brain imaging gets bigger, so there's some caveats. But nevertheless, if you think about, some people talk about this thing of brain reserve, and you build up brain reserve over your lifetime, and that protects you against Alzheimer's. Well, this doesn't disprove that, but it does suggest that you can't measure it with a brain scan, or at least not with any of the measures that we're using. So I will stop there, and we've got a few minutes for questions if anyone's got any. I think yes, just if you please hang on for the mic in case there's anyone online.
Thank you. You must have explained that well, because I understand, I think. One question that I was going to ask, would it be of any use then to do a similar scan with a remote tribe, or, something, because the discussion this morning was about the information we have is mainly from Europe, but I just wondered, like a really small tribe that have a different lifestyle?
Yeah, so these genetic variants are very, they really only hold true for the time and the place that you do the study in. And I think the best example for that is that currently the number one genetic hit for lung cancer is in the nicotinic receptor, right? So if we did that GWAS, and that, of course, is driven by the fact your nicotinic receptor drives smoking behavior. But, if you'd done that GWAS in Europe, but before people in Europe smoked, that would not have been important for lung cancer. So if you did a GWAS of some kind of proxy for education in a culture where there wasn't schooling, I don't know, you would have to come up with some kind of analogy, but some very different kind of schooling structure, then I suppose you might well get very different results. But if you were able to replicate across different cultures, then I suppose that would increase the confidence, both of your phenotypic, actually both of observational studies and of genetic studies, because by doing things in different cultures, you can sometimes, there's different confounders, aren't there, in different cultures. So if things are robust to many different cultures, then you can have more confidence in them. I don't know if there's any dementia clinicians or researchers here. It's my one and only paper on dementia, so people in the audience may know much more than me about it.
Thanks, Graham. So I have this personal theory that the brain is like a muscle, that you use it or lose it. So I think Alzheimer's Society and some private healthcare providers think that things like stimulation in the elderly population will ward off Alzheimer's, even if they haven't had very good education when they were younger. Is that worth thinking about?
Yeah, so I think that's part of this brain health drive, really, to think about. They think it's a useful kind of prevention strategy to sort of think about you build up your brain health just as you might build up your heart health. And it might be causal, mightn't it? But it's very difficult to know, because it might be that the same sort of people who do those kinds of things may be resilient, maybe the sort of people who are resilient to Alzheimer's through some unmeasured confounding. And the other thing I think that's interesting is that when I have presented to dementia clinicians, they say, well, right, so is Alzheimer's a clinical diagnosis? You have to have symptoms to have Alzheimer's, or is Alzheimer's a process of amyloid buildup in the brain, and you have Alzheimer's even if you don't have any symptoms? If you've got that pathological buildup, this is what the companies might be interested with these new drugs, right? For clearing amyloid. So does education stop the buildup of the amyloid somehow by predisposing you to be more active, and somehow that prevents the buildup? Or does it just mean you can kind of cope with it better? And so you, when the clinician asks you a memory test, you can somehow muddle through the memory test even in spite of the accruing amyloid. And you know, all of that, I mean, is very interesting as well. But in terms of advice, it's not going to do you any harm to, or it seems a plausible and sensible thing to do until we know the causality. But, I really don't think it's been causally demonstrated.
Yes, a quick one. I'm new to Mendelian randomization, so please bear with me. But I was wondering, on a meta perspective, what does this mean for future research? And I mean, nowadays the RCT is the golden standard. What do you think will happen?
Yeah, so I think you'll see. So I think Mendelian randomization, especially when it's something very closely related to a treatment target. So drug companies are very interested. You'll see some studies where there's a Mendelian randomization along with a clinical trial published, together. If you have a Mendelian randomization study that might suggest actually the phenotypic association is not causal. So that's been the case with in cardiology. Some of the molecules that in our blood are associated with poor cardiac outcomes have turned out just to be sort of epiphenomenal, not really causal. But if you don't know that, they might look like a very good target to treat. But if you can do a Mendelian randomization study and say, well, maybe this is actually, we don't have evidence for causality, you could save yourself the money of doing a trial. So I think if there's for example, you could do Alex's, if you do, whether it doesn't have to be a medicine treatment trial, you could think of brain activation, keeping fit or mentally active. If you do a big enough GWAS and then see if the Mendelian randomization, if that holds up as being potentially causal. I mean, it's a bit difficult to do a randomization about years of education and then measure 50 years later to see if you get dementia, but maybe you can do brain training in your 60s to see if you get dementia in your 70s. If you had enough confidence from the MR, maybe that might convince a funder to do a very expensive trial. So they're definitely very interested in these kind of techniques. So you can now do, especially now that there's a lot of good GWAS of proteins in the blood. So the drug companies can do a GWAS from those proteins in the blood, and they can see, look at that protein as an exposure and disease as an outcome and think, oh, if we upregulated or downregulated this protein, maybe it would be protective for a particular illness.
You showed the results here for the Mendelian randomization for Alzheimer's. Did you show it for schizophrenia as well, or would you do another study?
I haven't shown here one for. I've done some work on schizophrenia, looking at. If I mentioned schizophrenia, it was a slip of the tongue because I do so many studies on schizophrenia. We've done some work on smoking and schizophrenia, looking at that, and but we haven't looked at the education and schizophrenia. But I think sorry, did you have a specific question?
No, that was one thing, education and schizophrenia, or potentially number of acute episodes.
Yeah, well, I think that would be really interesting to look at. So at the moment, we just have GWAS for illness or not, don't we, really? That's what our phenotypes tend to be that go into these GWAS. But in the future, we want to have GWAS for things like number of episodes, good outcome, bad outcomes. And then, because once you have those GWAS, then you can do the Mendelian randomization. You can't do the Mendelian randomization until you have the GWAS. So we need to do really good studies with sort of the phenotypes that we're interested in. And then we could look to see, oh, here is something, yeah, okay, one thing is, do you get the illness or not? But, you know, some people have a really good outcome, don't they? And other people have a terrible outcome. So maybe knowing whether you get the illness or not is not so important. It's maybe looking within the illness. So there was some very nice work on COVID. So they did very rapid GWAS on COVID, GWAS of severe COVID versus mild COVID, and looking at what factors might be causal in whether you could have severe or mild COVID, but we haven't got those sort of equivalent data yet in psychiatry, but people are working on it.
So we could look at the Scandinavian cohorts retrospectively.
Yeah. So I think there is a lot of, you maybe already are doing this, are
No,
you?
no.
Yeah, no, there is. So in Finn, I think there is indeed. I think sometimes, like in FinGen, for example, there's very good genetic information, but you tend to have in hospital statistics, you know, diagnosis. What else can you get out of hospital statistics? Maybe you can get number of admissions. Yeah.
Hi, I'm just wondering, like, given all the research so far, are we going back to using Bleuler's concept of dementia precox as far as the schizophrenia is concerned?
Well, it's very. I think that's a big question. Maybe too big to answer in the short time that we have. Maybe we should discuss afterwards. Because we're slightly over time, I think.
I think we...
Yeah, well, let's stop and we can carry on in the break. But thank you very much, everybody.
Thank you again. Wonderful.